Precision medicine, I think, is a recognition of something that we've all known for a long time. That is, namely, we're not all the same. We're individuals. We're going to go back to the molecular level and think about people as individuals based on more a molecular profile. It's really going to fundamentally change the way that we practice medicine. Conventionally, doctors have grouped people by disease, say Crohn's disease, rheumatoid arthritis, heart disease, and so on. But if we zoom in and take what we've learned from studies like the Human Genome Project and the Human Microbiome Project and the research that these studies have enabled, we begin to see that within any disease group, there are subgroups that are on a molecular level very different from each other. It makes sense that if these groups have different molecular causes, then they will not respond the same way to one treatment. Now, if we zoom back out and look again at the same population, we discover that we have a very different picture. Across diseases, people share molecular signatures. Someone with Crohn's disease could be, on a molecular level, much more similar to a patient with a certain metabolic disease or rheumatologic condition than they are to other people with Crohn's disease. So now, armed with this new information, we can regroup people according to shared molecular profiles. And we may find that what is a very effective treatment for treating a subgroup of Crohn's can also be used to treat another person's rheumatoid arthritis. So as we dive deeper down into the genetics, the gene expression, the microbiome, all of that gives us information. You can actually discover one thing in one group of individuals, and it gives you great insight to diseases in other groups of individuals.
Well, microbiome is the collection of all the microbes in and on our body. The microbiome consists of the bacteria themselves and all the genes that those bacteria possess. The bacteria in our gut outnumber all the cells in our body, so we're more microbial of composition than we are human. We have a whole nother genome inside our bodies, which is the microbial genome. If we can start to look at our genetic makeup as the conglomeration of bacteria and human, it opens up a whole new opportunity for really understanding how we respond to our environment.
We're trying to capture the proteome, all of the proteins at any one time and any one moment in a person. The diagnosis that we make today, whether or not you're having a heart attack, is based on a protein that is circulating in your blood. We just want more of those proteins to be able to identify them. And the goal is to be able to take a drop of blood from your fingertip, a small amount. We put into our automated system that will get the protein out, digest it super accurately, target only those peptides we care about, and then we're able to look at your proteome. Always what we're looking for is the protein or the peptide that represents the biomarker that's unique, that tells us the status of you at that moment.
In terms of not just genetics and microbiome and so on, we think about near-patient technologies. Once we do get people on the right therapy, can we monitor them at home using smart technology?
Once we start treating patients, we have to precisely know if it's even working. And there's a whole science to this. We call it outcomes research, literally measuring the outcomes of the care. We can measure how somebody feels precisely. We can monitor patients remotely with biosensors. We can use wearable sensors on them and measure how well they're getting through the day. Are they getting up and down stairs? Are they walking? What's their heart rate? We can measure biomarkers, psychomarkers, and we can measure wearable data and triangulate all three of those data streams to understand precisely whether a treatment is working or not in an individual patient.
At Cedars, we are embracing the big data approach to precision medicine.
Bioinformatics is the study of large-scale data sets that are generated within the biosciences, and that could be either on the biology side, molecular sequencing, or imaging, and on the medical side, that could be electronic medical records or information collected from mobile devices. We deal with data sets that are up to a petabyte, which is massive. Analyzing the genome, for instance, of a particular patient would take a year on a single computer. So we need to basically spread it out over hundreds of different servers to get it done within a reasonable time frame. And for that, we need these large data centers with thousands of individual servers that we can use. We have a fantastic infrastructure at Cedars-Sinai that has been built built up, over the last several years in concert with our switch to digital electronic medical records. We really need to be able to integrate this molecular DNA data with the electronic medical records to really be able to figure out which genes are affecting patients in different ways.
There are examples that precision medicine is already happening. You will have heard about sequencing the tumors.
We have the ability to find genetic variants which are in a person's DNA blueprint that say, this means you are more likely than the person next to you to get, for example, breast cancer or prostate cancer. And we can therefore use this sort of information to say, well, if we know you're more likely to get prostate or breast or colorectal or ovarian cancer, can we do something about that? Can we clinically intervene to stop you getting cancer? And that is part of a massive interaction that's going to develop over the coming years between the people who can look at every one of those 3,000 million DNA letters in every individual who comes through Cedars, and the clinical side that will be able to say, well, now we know this is what your personal profile is all about, this is what we're going to do about that. None of this can really happen without having an incredibly good clinical setup.
So the Biobank is an ongoing project. We have collected already thousands of samples, but we need to collect many more so that we get a complete understanding of each disease. Collecting all these samples will allow us to study the differences of diseases across populations. From there, samples go to the laboratory. We stain tissues with 7 different colors. We can use the computer to separate and analyze those colors in each cell in the tissue. We can analyze how much of a given pattern exists in the image. This allows us to determine the difference in the disease between individual patients.
One of the philosophies of the Precision Medicine Initiative at Cedars is to work with partners in industry to make sure that we have access to the best technology. We're right at the front of the line when new technologies become available.
We really want to bring the newest medicine, the newest treatments, the best technology to benefit our patients as quickly as possible. To do that, we need the best clinicians, which we have. We need the best scientists, which we have. And there's a third part to that, which is really bringing our contacts from industry. We want to ensure that we have access to those technologies. We want to make sure that we are funded so that we can provide these new techniques. We also want to make sure that we play a role in developing the newest technologies as well. If we can produce great technology at the lowest possible cost, then it means we can treat more patients quicker. And that has a great benefit. So many things are coming together at the right time to create what I think is the most exciting time in science and medicine.
Precision medicine is going to be a disruptive force in medicine, but it's going to be a positive disruptive force. We've faced these before, whether it be the invention of the stethoscope or X-rays or CT scans or MRIs, and all of these things enhance clinical care.